Hello there, student entomologists. If you're looking for a fun yet insightful laboratory activities that will not just fuel your passion for entomology but will leave you with great knowledge, then you're lucky, as we are going to walk you through with the must-knows and must-have skills of a student entomologist. So prepare your materials and let's get started.
To start off this presentation, let me first discuss the first laboratory exercise, which is the insect external morphology. The objectives of this exercise is to, firstly, be familiar with insect morphology. Secondly, know the basic knowledge of entomology. And lastly, be able to distinguish insect body parts. This video from Soybug is part of the module 1. The video is about how to look at an insect. It covers how to view the insect from different angles, such as lateral view, which is the most important one, because it is also called the profile view or the view from the side. The dorsal, ventral, posterior, and also the anterior, which is the face-to-face view, like you're having a staring contest with them. It again discussed the three major segments of the insect. Additionally, the video details the different types of antennae and their functions. Subsequently, we were tasked with image collection, searching for relevant images of insects from various sources such as PDFs, online databases, journals, and also ebooks. What you are seeing right now is a collage pictures of the drawings using the images from the previous slides as a reference. And aside from drawing the needed insect external body parts, the group were also tasked to view some prepared slides and draw them to aid on being more familiar with them.
For laboratory exercise number 2, its main objective is to identify and know the different collection tools for arthropod collection. The different equipment include: first, the basic equipment, which includes forceps that is used to grasp specimen with care, vials that contain the preservatives, killing bottle, which is essential in killing the specimens used in studies that contains the liquid killing agents composed of ethyl acetate, ether, chloroform, and ammonia water. We also have small boxes or containers for storing specimens, envelopes for temporary storage of delicate specimens, aspirators for collecting small insects at night. We also have tissue required for the killing bottle and aspirators, notebook for labeling and jotting down notes, knife and a pair of scissors, which is essential in cutting open galls or twigs, fine brush for picking up minute specimens, and a hand lens. Second, we have collecting nets, which is divided in three basic forms, which are aerial, sweeping, and aquatic. However, in our case, we only needed the first two forms. Aerial is designed to collect butterflies and other flying insects, while sweeping is similar to aerial but has a stronger and more durable bag to withstand being dragged through dense vegetation. Lastly are baits, lures, and other attractants, which are used to attract insects through the use of baits.
In this laboratory exercise, our objectives are to collect insects within the campus vicinity through the two methods, namely the active collection, where we collect insects by hand or through a sweep net, and in passive collection, where we place baits in a particular location and wait until insects come. Furthermore, the collected insects will be processed and tallied to determine its abundance and diversity on the location chosen. So to start the collection proper, we have prepared the following materials, such as the sweep net, killing jar, fine brush, tubes with alcohol, ziplocs, baits, and alcohol spray. The killing jar was used to euthanize the captured insects through the use of ethyl acetate, which ensures that the insect's body remains intact and undamaged, making it suitable for preservation and study. On the other hand, the alcohol spray acts as a stunning tool to slow down the motility of the insects, enabling easy collection. Our baits consist of a mixture of dead spider and flies, as well as peanut butter and tuna. This kind of mixture will generate a more generalized and diverse bait that could affect a wider or greater range of insects. The chosen location was the education building of FE Manila, wherein baits were placed both indoors and outdoors. The baits location's elevation and GPS coordinates were also recorded.
While collecting specimens around the educational building, we employed two sampling methods. First, we conducted passive sampling, where we set up traps to catch ants both inside and outside the campus. While waiting for results, some group members engage in active sampling, which involve hand-picking ants and placing them in tubes filled with alcohol. Additionally, we use aerial nets to capture butterflies and flies. So these are the specimen that we gather around the educational building. Lastly, we conducted a tally matrix from the collection proper, and these are the results.
Now let's proceed for the lab exercise 4, which is entitled Basic Arthropod Mounting for Identification. So the main objective of this laboratory exercise is to successfully mount the specimens we collected from the previous laboratory exercise. So this is essential in observing and determining characteristics of different arthropods and for storage as well. Presented on the screen is the essential materials for mounting a specimen, such as entomological pins, pinning block, entomological puncher, and paper tips, water-based glue, which is the tight bond, a Gucci ball, which is basically a ping pong ball with cement, and clay sitting on a plastic bottle cup, and entomological probe to be used in manipulating the specimen, and of course a dissecting microscope.
Here in the video, Doc Pag-ong is assessing the specimen that he will be demonstrating to our group, which was a suspected queen because of the presence of its enlarged thorax. Well, as we can see in this video, Doc Pag-ong is already conducting the demonstration of how to properly mount a specimen. Shown on the screen is the mounted specimens of the members in our group during our practical examinations. One of the important takeaways we got from this lab exercise is the importance of precision when it comes to mounting specimens for us to identify them more particular and accurately. It is also important to take note that when mounting a specimen, we should have the right and functional materials, specifically entomological probes, that is used in manipulating the orientation of the specimen. It is also important to take note the placement and orientation of the specimen, and of course, the glue integrity.
In Laboratory 5, the group answered the following questions with regards to the research paper of Leb et al. 2014, which is entitled Mosquitoes and the Relevance as Disease Vectors in the City of Vienna, Austria. So we answered the scientific gap and what is the research all about. Lastly, we also watched a video by Eric Westus entitled Mosquito Blues, a tiny fly with a big impact on human longevity. In Laboratory 6, the group identified and enumerated all the countries that are affected with the malaria vector, and we also listed down all those countries that does not have any vectors. Overall, we observed that the sub-Saharan African region are the ones with a lot of malarial vector, and up until now, they are the leading countries with increasing death rate and mortality rate. In laboratory exercise 7, the group identified the differences between the larvae of Culicine and Anopheline mosquitoes. So the Culicine mosquitoes has no palmate hairs, it has an air tube, and it lies at an angle to the surface of the water. While on the contrary, the Anopheline larvae has palmate hairs, it does not have an air tube, and it lies parallel to the water surface.
Laboratory 8, entitled Black Flies, combines the analysis of the paper biodiversity, seasonal abundance, and distribution of black flies in 6 different regions of Thailand, and the video analysis of how to protect yourself in nature. Black flies in Thailand exhibit regional variation in their distribution and abundance. Northern regions are more conducive to a higher diversity of species, particularly during colder seasons. Conversely, southern regions favor black flies adapted to warmer conditions. And the video discusses on how to protect yourself by using different insect repellents, wearing lighter clothing, and applying eucalyptus-based off lotion. Laboratory 9, entitled Sandflies, focuses on the analysis and transcribing of the video surveying sandfly breeding habitats, where it mentioned the importance of studying, identifying, and using different laboratory methods in order to study sandflies. Aside from that, it also focuses on the identification of different parts of mosquito, knowing the collected mosquito subfamily and genera, and dissecting its proboscis and trying to fish out its salivary glands. We were able to accomplish everything from the dissection of proboscis, where we identified its labium, proboscis itself, and the palps, as well as showing that it was female due to its pilose antennae, and it belongs to the subfamily Culicidae and the genera Culex.
And that's all for our group presentation of laboratory exercises 1 to 9. I can tell we've gained valuable insights into their biology, breeding habits, and their potential health risks. By understanding them, we can contribute to developing effective control measures for the diseases they bring. We hope this presentation has been informative and has sparked your interest in the field of entomology. Thank you for listening.